There we go. Uh, Welcome everyone once again to our Los Angeles Birders webinar for October 8th. We have a wonderful program in store, but before we get to that, I just want to remind you, if you have any questions on that program, please pop it into the Q&A and our presenter will get to you and it'll help us keep track of everything, keep it in order. If you're a member of Lab, thank you very much. I really, and we really appreciate it. If you're not a member, please consider becoming a member. It's very inexpensive, and it allows you to interact on these webinars with Q&A and with polls when our polls are up and running. Lab members get first dibs to our field trips. We have lots of student learning opportunities. We have science opportunities. And plus, you get the warm and fuzzy feeling of supporting a wonderful organization like Los Angeles Birders. So please support us. And with that, I'm waiting for there we go. With that, I'd like to introduce Molly Hill, who will introduce our speaker for tonight. Molly?
Thanks, Ron. It is my pleasure to introduce today's speaker, Dr. Myra Finkelstein, who will be sharing some groundbreaking research on one of North America's most iconic species, the California condor. Dr. Finkelstein is part of a dedicated team of conservation biologists that have been working with over 20 years of data on reintroduced California condor flocks. Her team's latest research, published in Biological Conservation, uses advanced modeling to predict the future of condor populations under different management strategies. In their findings, they've identified a remarkable pathway to potentially transition the condor from being a conservation-reliant species to one that can thrive independently in the wild. Today, Dr. Finkelstein will discuss how this could be achieved through a single key action, the societal shift to non-lead ammunition, and share insights on maximizing captive breeding efforts, mitigating adult mortality, and preparing for catastrophic threats like wildfires or disease. Dr. Finkelstein will also give a quick summary of her latest research with another iconic bird, this time of New Zealand, the kea. Without further ado, please join me in welcoming Dr. Myra Finkelstein.
Okay, thank you, everybody. I guess I should start sharing then.
Yes.
And there we go. So, this works. Okay, can you see it?
Yes, that's up and running.
Okay, thank you. Thanks for inviting me. I'm honored to be here today. Thank you so much for the introduction, Molly, and I'm excited to learn more about what you do with kea. All right, I'm gonna just go through my research, and then if you have questions, that's right, you put it in the Q&A, and I will try. I can answer. I can pause. I have like little vignettes, so I'll pause after each one, and we can answer questions as we go. So outline for tonight, I'm going to do an opening act talking a little bit about my my newly published, hot, still on the press work with kea in New Zealand, sources of lead in kea. Then I'm going to talk about condors, a little bit of a brief history and their current status, our work on models to inform management, and then some of the future work that I'm doing currently right now on lead exposure and risk and how it's very complicated. So for the opening act, the title of our paper that's in press right now in Conservation Letters is Lead-Based Ammunition as a Threat to the Endangered New Zealand Kea. So I was contacted by researchers and people that work for the Department of Conservation in New Zealand because they were really concerned about kea and their lead poisoning. And they are an endangered species and they're documented to have high blood lead levels. So the current hypothesis for why these kea are lead poisoned is that they eat this lead-containing building materials on these huts, these old huts that people go to and hike to and things like that. But another worry is that they're also exposed to lead-based ammunition. So Kia have been observed scavenging on carcasses, and we know that some of these carcasses that they're observed scavenging on has been shot with lead-based ammunition, because the Department of Conservation still uses lead-based ammunition in their hunt and culling the non-native mammals in New Zealand, and then the Kia will eat feed on these carcasses. So I was contacted because although there is current work being done to help mitigate the lead exposure from these huts and these building materials on these huts, there really was not a lot of work done to try to understand how lead ammunition could be affecting them. And there's a lot of resistance to trying to do something about the lead ammunition problem. So they needed information. So the goal of the study was to do lead isotopic compositions of Kia blood samples, ammunition that are from New Zealand, so that's been used in these hunting and culling programs, and also lead-based building materials. So primarily they're flashing, there's the roof flashing and roof nails are been made with lead. And so basically looking at these three items to identify sources of lead to Kia. So first, to talk about the lead sources. So before we even started this work, or actually really got into it, we looked and found the isotopic composition of lead-based building materials, such as flashing and nail heads, that was sent to me. So these were taken from the huts in New Zealand, and they have an isotopic signature here. And they're all fairly similar. These are the nails. They look similar with the building materials. And then if you contrast that with the ammunition samples that I received from New Zealand, looked at ball bearings, buckshots, and bullets, and you can see they have a signature that's lower isotopically than the lead-based building materials, and importantly, non-overlapping and measurably different. So can you see my cursor, I hope? Maybe, yes? I hope.
I'm sorry, I didn't hit the mute button.
Yes.
Yes, we can see the cursor.
Okay, good. So here in this corner here is our measurement error, isotopic measurement error. So we have a really pretty tight precision for measuring isotopes. So you can see that definitely these samples are measurably different than these samples. So that was the first thing that was important to say. So I basically told the team in New Zealand, yes, there's a difference between the isotopic signature of lead in the huts and lead in the ammunition. So send the blood samples over so we can see if they were exposed to lead from huts or ammunition. And that looks like this. So on the y-axis here is the isotopic ratio, and the x-axis is blood lead concentration. And these are the Kia samples that we analyzed. And you can see the green ones through probability modeling have an isotopic signature that is not different than the huts. So they look like their lead in their blood looks like the lead in the hut. The gray ones are ones that couldn't be determined either way. And the blue ones, the lead in the blood looks like the lead in the ammunition. So the conclusion of this work, which is it's kind of simple in a way, is that about 80% of the birds could be identified as being exposed to either huts or ammunition. So about 40% to huts, 40% to ammunition, and the remaining percent were undetermined. But if you look at the undetermined, they basically look like a mixture between the huts and the ammunition. And my most likely hypothesis is that's what they are. They're a mixture of both. So this was really important to show that, yes, Kia, the isotopic composition looks like the source of lead exposure is from ammunition. Huts as well. Huts is a problem, but so is the ammunition. We also found regional differences between sources of Kia lead exposure. So in general, here is a map of New Zealand, and the little icons are all the blood samples that we took. The huts are the birds that had an isotopic signature that looked like hut, and the ammunition is an isotopic signature that looked like ammunition. And this is when the birds were sampled. So in general, Kia exposed to building materials were captured near the huts, near those building materials, and Kia exposed to ammunition were captured kind of where hunting occurs and away from huts, kind of where we think these operations occur. So this is just more evidence showing that these Kia are exposed to these two different sources. So to summarize this little kind of quick summary, we found that the lead isotopic compositions indicate that lead-based building materials and lead-based ammunition are responsible for the majority of lead exposure in the Kia, with about equal numbers of Kia being exposed to each. So this was really important finding, and our partners with the Department of Conservation were very anxious and excited to get it published, because this publication is now being used to help basically advocate for the switch to non-lead ammunition, especially in any government-sponsored operations in New Zealand. So this was a really rewarding project for me because I felt like it had very direct conservation benefit and potential policy implications. So that's the Kia little overview. I don't know if anybody has a quick question for that now, or if we should just move through to the end and ask questions.
Yeah, any questions you can pop down in the Q&A. And the only question we have in there at the moment is about condors.
Okay, well, let's move to condors. So California condors. I've given a talk on condors before, so I'm just giving I'll give a little overview. They're North America's largest terrestrial bird. They can have a wingspan of about 9 feet, weigh about 20 pounds. They should be able to live about 70 years. They start breeding at about 6 years. They typically have one chick every other year. So they're a very K-selected species. They evolved to have low reproduction over a long lifespan. I wanted to shout out because I talk about this work and it's me just sitting here on the Zoom, but I have a huge amount of people that work with me. And in particular, the heavy lifting of all the modeling work I'm going to show you is Dr. Victoria Bacher. So she's the quantitative ecologist that I work with. And, I always say that I know enough about modeling to be dangerous, but really she's the one that is the modeling genius behind the work I'm going to talk to you today, plus a bunch of other people. So I'm the messenger, but I am no means, the only one that has pushed this forward. So I wanted to just now talk a little bit about the brief history and current status to say, kind of set up for some of the models that we've just published. So California condors have had a rapid decline over the past 200 years. And about the end of the 1800s, there was believed to be about 600 birds. But in 1982, there was only 22 condors left, and it's this little U shape down in Southern California, the very small range. And the 1800s, I should show up, is this whole big gray area we thought that they were in. They're one of the world's rarest birds. They were listed as federally protected since the 1970s, and the IUCN lists them as critically endangered since 1994. In the 1980s, they started a captive breeding program. In 1987, because condors were still dying at alarming rates in the wild, they made the very controversial decision to bring them all into captivity so that they could maximize the genetic pool that they had in the captive breeding program, since the condors weren't living long enough to breed in the wild. So they brought them all in. And then in 1992 was the first release of these captive-reared birds back into the wild. So in general, the captive breeding program has been a huge success. California condors, we do status reports every year. So at the end of 2023, the total population in the world was 561. In the wild, we had 195 in California. Well, including then if you include the Northern California, which is so new. I don't always think of it right in my head. There's 195 in central and southern, plus 11 in northern now. We have 48 in Baja, and then 90 in Arizona, Utah. I'm going to talk to you about these models today. We've worked primarily with the California population. We actually are doing a huge global population model right now. That work is not published and it's still kind of being put together. So maybe the next time in the next year or two, I'll tell you about the global model. But today I'm just talking to you about the California model with the California Condors in the established release sites. So in Pinnacles, Ventana, and Bitter Creek. So we don't have the Northern California in these models population. So unfortunately, our new models show that California Condor population growth remains reliant on captive bred releases. And without releases, the wild California Condor population would be declining. So this is like a population viability analysis model. So on the y-axis is total population size, and then you go year. So the dots here are observed, and then these data kind of are from 2018 onward. So we observed 2018, and then we start projecting based on these observed rates. And with releases, the population is growing, but without releases, the population is declining. And let's see if this is going to work for me. Yeah. So in the red line, I'm just showing you, so population of 1 is stable. So it's not increasing or decreasing, but you can see increasing with releases. Unfortunately, it's decreasing without releases. So that's a bummer. Yeah. So what do we do about that? So how can our models help inform management, recognizing that the California population is still reliant on captive bred releases? I'm going to talk about maximizing this captive breeding resources, offsetting adult mortality, and a little bit about how to buffer against catastrophic events. So our work with the Fish and Wildlife Service is to try to help build models to inform species recovery. And in general, you have policy and management actions that are kind of put forth to help populations grow. You also have, for wild populations, threats to survival and reproduction, which impact population health. And then you have data that you've collected to understand what the current status is. So the model I just showed you showing the current status is declining, that's all based on observational data and other types of monitoring data. And then with the data, you build these PVAs, population viability analysis models, that help predict what's happening in the future to inform these decisions and actions. And typically, you talk to managers, you look at the threats, you gather up your data, and you build a model, and you're done. And you're like, okay, we're done. But there's a new kind of way of thinking about this, which is called population viability management, where you're not done just after you build the model. You work then with policy and management to say, well, how did those actions then affect the threats and affect population health, like survival or reproduction? So you kind of build these iterative PVAs to kind of help kind of move forward, because it's not just a static process. So we've done this for California condors, and like everything California condor, and it's probably because we have so much data on condors, it's not quite so simple. So I'm just going to give you a little overview of what our population viability management analysis looks like for condors. And so we have different conservation actions, like replacing failed eggs, managing trash at nests, releasing captive bred juveniles, reducing lead mortality through non-lead ammunition policy, education, vaccination for diseases. And then we have the efficacy of those actions, like, what's the fledge rate of the bird with and without trash management? What's the survival rate? What's the lead mortality rate? And then we have these different threats. We have DDE, which reduces hatch success, trash, which reduces fledge success, lead, which reduces free-flying condors and nestling survival. We have other anthropogenic potential mortality, like wind farms, and these new emerging catastrophic threats. And all that feeds into understanding survival and reproduction and abundance. And then we do demographic rate monitoring, abundance monitoring, planning. We put in observation error. And then we do these iterative PVAs where we basically keep kind of working with the stakeholders, aka primarily the Fish and Wildlife Service, to try to help understand what's working and what's not working. So it's not this kind of one-time thing. So this is a little bit more detailed of the way this modeling approach works. So as I showed, the California population is growing due to captive bred releases. So the first thing we wanted to look at was, well, can we help understand how to maximize these captive breeding resources? Because it's not cheap. Yeah. So early in the program, they started this sort of policy of checking the nests, and if the eggs were failed, they would swap out the failed eggs with zoo-laid eggs. This was done across, like in Southern California as well as Central California, but it was done a lot in Central California where they'd swap out the eggs even if they weren't failed because they were worried about eggshell thinning and from DDE. So these are all collaborators had published this work showing that evidence of eggshell thinning and lower hatching success in condors on the coast related to DDE exposure. And we found this too in our modeling. On the y-axis here, these are our models. Probability of an egg hatching is basically lower with the higher probability or the higher amount of DD exposure that the condor has. So based on kind of the wanting to have as many, successful eggs in the nest, so swapping out failed eggs regardless, also the worry of thin eggs in Central California, there's this like kind of major effort of checking every nest, candling the eggs, and if they failed, replacing with a dummy, or if it was dead, you put a dummy egg in there. Then at the right time, you have a zoo-laid egg that you swap in sort of right before it hatches. And it's a lot of work. So we wanted to understand, is that a good idea? Should you prioritize nest management or should you prioritize captive bred releases? Because again, these eggs that are swapped are zoo-laid eggs. You could let them grow up a little bit in the zoo, or you could swap out that egg in the nest. So we found kind of overwhelmingly that the releases of juveniles increases population growth more than replacing eggs in wild nests. And on the y-axis is the number of eggs replaced. On the x-axis is the number of juveniles released. And each kind of contour band is a different growth rate. And the growth rate as you go from red to blue gets higher. So you're kind of going up, 1% in growth rate with each band. But if you look here, if you have 5 juveniles released, you're in the band basically right up to like 1.01 growth rate. And that corresponds, so this is 5, that corresponds to 10 eggs replaced. And so across a broad range of mortality conditions, the release of a captive bred juvenile had about double the effect on the stochastic growth rate, so this kind of what the rate is, compared to the use of an egg laid in captivity to replace a failed wild egg. So you get a ton more bang for your buck if you release those juveniles and you leave those eggs to basically, they can fail in the nest, and then you take that zoo-laid egg and you raise it till it's 1.5 years old, and then you release the bird. Because of this work, Fish and Wildlife Service actually changed their management policy, and they stopped pretty much overwhelmingly replacing the failed eggs, and they started to try to up the number of captive juveniles released instead. So unfortunately, lead poisoning remains the #1 mortality factor for adult condors. And so we've talked a lot about captive breeding to offset lead mortality. And there's a lot of kind of talk with Fish and Wildlife Service. Well, can we just release more birds? And how many captive birds do we need to release to kind of make up for the lead mortality? And so just sort of trying to get around that. And so in our paper, we looked at a couple of different ways to do that. I'm going to highlight one offset that we talk about today, just for time's sake and simplicity's sake. And that one, I think, is really important, which is showing that reducing lead mortality is much more effective than releasing juveniles in terms of having, helping long-term population growth. So for the graph here, so this is a starting population size of 100. On the y-axis is how many juveniles are released. And then the x-axis is lead mortality, like percent lead mortality. And this would be like 1%, 2%, 3%. And again, you have these contour lines that represent the growth rate, and each band is about 1% growth. And so we found that basically 1% reduction in lead mortality, if the population size is 100, is equal to about 3 releases for starting population size of 100. And what this looks like is if you kind of switch, so you're reducing lead mortality from 0.02 to 0.01, you cross a whole band. So you basically gain a whole percent in growth rate. And to cross a whole band for captive juveniles, it's about 3. So you can see here that, yeah, it's much more effective to reduce mortality than to release more juveniles. But the thing that's so interesting about this is it's kind of like a compounding benefit. So if the population is at 300, 1% reduction in lead mortality is equal to close to 7 releases for starting population size. So again, on the x-axis is lead mortality, and 1% is about a band. So that's 1% lead mortality. If you reduce like 1.5 to 0.5, you increase your growth rate by 1%. But this band to go up 1% for captive juveniles released is close to 7, 8 birds. And this is the whole thing about compounding interest. And so we've done this work, and other work, which we really try to show that we cannot like captive release our way out of this problem. Like we have to reduce lead related mortality. It's not going to work. It doesn't matter how many juveniles you release out there, especially if the population growth rate is less than 1, like it is now. You're going to be doing it forever. And this really shows how much more bang for your buck you can get by really reducing the lead mortality. And you might think, okay, well, that makes sense. And why, that doesn't seem like that's such a, you might think that, well, that's intuitive, that makes sense. Why do you have to explain it like that? But if we're going to, for example, like the Fish and Wildlife Service, PG&E, and they're saying you killed so many X number of condors, hit your power lines and died, and we need you to pay that back to the Condor program. So we don't want you to pay it back by giving us more captive juvenile released. Look what you can do. You can help us basically lower lead related mortality. And that's such a better thing to do. And that's actually what's happening right now. They're negotiating with Pacific Gas and Electric, PG&E, to try to offset some of the mortality that we've had from condors by hitting their power lines and their infrastructure, and using models like these to show, instead of just more captive juveniles released, try to help us lower lead-related mortality. So condors have faced 2 catastrophic events over the past 4 years. In 2020, we'd had 12 condors die. About 10% of the Central California flock were killed from a wildland fire that went through their nesting and roosting habitat. 3 years later, we had 21 condors confirmed or suspected of dying from highly pathogenic avian influenza. 20% of the southern flock. I mean, it was a huge loss. So given these two catastrophic events over the past 4 years, we realized we really needed to start incorporating these catastrophes into our models, trying to predict what's life going to be like for condors in the future. So we found, not surprising, that on the y-axis is total population size. Now I'm just showing you the one without ongoing releases here, that they can't withstand frequent catastrophic events. So again, the red line across would be what would look like if the population is stable. Currently, without releases, the population is not stable. And then you can just model in catastrophes happening every 8 years or 4 years or 2 years, and taking out, this is taking out 20% of the population, but you could take out 10%, and sort of showing then what could happen. And it's underscoring that basically this, like condors, the population is too small, the growth rate's too low, can't handle it. And the thing that we think this is important right now is we've really now, with our models moving forward, are simulating these catastrophic events into the models. So all the management planning that's going forward is taking into account that these can happen. And since we're simulating them, we could do them at like 8 years or 4 years and different kinds of percent of the population. So we have flexibility, but we're able to say, hey, we need to account for that. We need to buffer the population against these additional threats that we know can happen based on the last 4 years. So to summarize these model results, unfortunately, the California condor population growth is still reliant on captive bred releases, and it's primarily due to lead-related mortality. That's the #1 mortality factor still. Releasing captive bred juveniles results in higher population growth and nest management. As I said, Fish and Wildlife Service has already implemented a change to basically try to divert resources from checking eggs and replacing eggs into releasing more juveniles. We're illustrating how small reductions in lead-related mortality outperform the ongoing releases of captive bred juveniles. And again, that work is being used to help try to advocate for mitigation from companies like PG&E to help provide resources such as non-lead ammunition to offset lead-related mortality. And just underscoring that condors do not have a high enough population size or growth rate to withstand these frequent catastrophic events. So I'm wondering, I could take questions now, or I can go to my last part, which I was not going to maybe have recorded, which is what I'm calling here the elephant in the room.
Okay, I'll stop the recording now. We can move on. And
Okay.
we have a couple of questions waiting, but we can hit everything together. Let me stop the recording.